All right, this is the Chapter 10 video. We're going to talk about biotechnology and recombinant DNA technology. Biotechnology is the use of microorganisms, cells, or cell components to make a product, and that product is often a protein. We could use biotechnology to manufacture foods, antibiotics, vitamins, enzymes, and other medicines such as insulin. In order to complete a biotechnology process, you have to use recombinant DNA technology, which is the insertion or modification of a gene to produce a desired protein. To do recombinant DNA technology, you need a vector, which is a self-replicating DNA used to carry the desired gene into a cell. So if I want to manufacture or use a cell to produce insulin, for example, I need to use a plasmid with the gene of interest, the insulin gene, to insert that into the bacteria, so the bacteria then creates the protein insulin. Viruses can also be used as vectors. The first step in a recombinant DNA technology process is extracting the DNA. We need a source of DNA to manipulate it for the biotech application. So it's the first step in the process. We can extract DNA by disrupting the cell wall or cell membrane, depending on the type of cell that we're actually interested in. And we can do this either chemically and physically, and a means to free the DNA. Once you have the DNA extracted, you want to make multiple copies of that DNA. So we use this process called polymerase chain reaction. It's just a method used to amplify or replicate DNA in the laboratory. So we can amplify to detectable levels, we can sequence the DNA, we can detect pathogens. There's all types of different applications for polymerase chain reaction, or PCR. The target region for any gene of interest is usually a short portion of a gene. However, we can amplify the entire gene as well. It's a very fast process, and we can complete the PCR reaction in about 2 to 3 hours, depending on the application. What we need to do is we need to use primers, which are used to selectively replicate only the target region. So these primers are short sequences of complementary DNA to the gene of interest. I'll show you a video here in a moment, and it will illustrate what the primers are. The primers provide a starting point for the DNA polymerase to attach nucleotides to. So it provides a 3' end, very much like our primers for DNA replication in the cell do as well. And PCR is possible due to an enzyme called Taq polymerase. Taq polymerase is a heat-stable DNA polymerase from a thermophile called Thermus aquaticus. This is important because our first step in PCR is to denature the DNA and heat our DNA to 95 degrees Celsius. At that temperature, the double-stranded DNA the hydrogen bonds will break. So we're going to make single-stranded template DNA. So if you remember from the DNA replication video, there's an enzyme called helicase inside cells that will separate the double-stranded DNA. In PCR, we are using heat. There is not an enzyme to break the double-stranded DNA. Heat will do that. Some medical applications of PCR include confirming diagnosis or identifying organisms causing disease. In the future, I would expect more and more diagnosis to be in a biotechnology application such as PCR. One example would be Bordetella pertussis, and this is done now in order to confirm a patient has pertussis. Pertussis is very difficult to isolate due to its extreme sensitivity to desiccation, so it dries out very easily. So we can have primers specific for target regions on the Bordetella pertussis genome and use PCR to amplify pertussis genes. The presence of the genes specific for that organism, Bordetella, would confirm the patient does indeed have pertussis. Some other applications of PCR products or the amplified DNA would be to use a restriction enzyme. and that allows scientists to cut DNA into fragments. A restriction enzyme will recognize a DNA sequence, usually a 4 to 6 base pair nucleotide sequence, and it will cut the DNA at that sequence. So here are some examples of some restriction enzymes. BAMH1, for example, will recognize the sequence GGATCC. And where it sees the G's here, these arrows indicate where it's going to cut. But it doesn't cut straight through for this. It cuts down through the G's and then across and then down. So this creates what we call sticky ends. Sticky ends here, as illustrated with this restriction enzyme, allow scientists to insert genes into this sequence then. Since we have single-stranded DNA, as long as the gene of interest has a TTAA at each end, or an AATT at that end, we can insert the gene and use that for recombinant DNA techniques. Hey3 here, for example, recognizes GGCC. So where the enzyme sees the GGCC, it will cut straight through. And that creates what are called blunt ends. Blunt ends here, as illustrated in this image, would be more for an application of DNA fingerprinting. We'll talk about DNA fingerprinting here in a moment. The restriction enzyme application, DNA fingerprinting, uniquely identifies individuals based on DNA fragment length. Fragments are generated by the restriction enzymes, and each individual's DNA is different enough that these enzymes will generate different lengths or fragments for two different individuals. In order to actually view the DNA fingerprints, we use a process called gel electrophoresis, which is a way to separate DNA fragments based on their base pair length. So what we do is we load DNA into a gel matrix, which is it's called an agarose gel, and we apply an electric current to the chamber. Smaller fragments will travel faster through the gel, and we create a banding pattern of fragments. Since DNA is negatively charged, basically the DNA will migrate to the opposite, so it'll migrate towards the positive anode. So this image here shows what I was mentioning here. We have these little wells here that we load the DNA samples into the gel, and we apply the current, and the bands will separate based on their number of base pairs. The shorter ones go farther through. This is what the gel actually looks like. We actually use this chemical called ethidium bromide to attach to the DNA fragments, and it will fluoresce under UV light. In order to explain the how shorter fragments migrate farther, I always tell students to imagine holding hands with a group of people and trying to walk through a cornfield. If you have 5 people holding hands, you're going to be able to migrate or travel much faster through the cornfield, that matrix, rather than a group of like 25 people, for example. Again, here's another gel image. You can actually see the wells here at the top that are created, and here on the left-hand side, we have what is called a molecular base pair standard, or a 100 base pair ladder. So at the top here, this is actually 1,000 base pairs. This is 900, 800, 7, 6, 5, 4, 3, 2, and 100 base pairs. So using the molecular base pair standard, we can compare the fragments here and estimate the size of the fragments based on this molecular base pair standard. So this fragment here at the top is about 1,000 base pairs, where this one down here is 9, 8, 7, 6, about 650 base pairs, because it falls right about in between the 6 and 700 base pair marker. So we can apply DNA fingerprinting to numerous applications. So in a crime case, just for an example, here in lane 1, we would have a victim's DNA. Lane 2, potentially skin samples from under that victim's fingernails, and we can compare suspected individuals to these lanes to find out who potentially was at that crime scene. So we compare across, so lane 4 matches up with lane 2, which were the skin cells found under the victim's fingernails. So that person 2 is likely to have been at that crime scene and potentially was involved in the crime. So this blue box or gray box represents the actual gel. The black lines represent the DNA fragments. The DNA is negatively charged, so it migrates down to the positive end. Who was the potential murderer in this case? We would say most likely this person too. Another application of using restriction enzymes would be to genetically engineer prokaryotes. You can do this with eukaryotic organisms as well. Using a restriction enzyme that recognizes a specific sequence, we can cut the plasmid DNA, so indicated over here, cut that plasmid and insert the insulin gene into the plasmid. We do a process of transformation, which is the uptake of cell-free DNA from a cell's environment. The cell now has the plasmid, and that cell will then produce that protein insulin. If you know somebody who has diabetes and uses insulin, the label would say that it is produced using recombinant DNA origin. So it is structurally similar, or identical, to what is produced by the human pancreas. And in this case, it was produced using the organism E. coli. So E. coli produces that insulin. Yeast cells can do this as well. We harvest that insulin, purify it, and give it to patients. Another application would be probe technologies. And what happens, or what we do with this, is we can use what are called DNA probes, which are used to locate specific nucleotide sequences of DNA or RNA. So single-stranded DNA or RNA has a fluorescent dye attached to it, and that can attach to a nucleotide sequence. The application of this is called FISH, fluorescence in situ hybridization, which allows us to rapidly identify organisms bypassing the actual need to grow the organism in the culture. So this would be important for organisms like Mycobacterium tuberculosis. We can take a sputum sample, mix the sample with specific probes for tuberculosis, and if we have amplification using fluorescence microscopy, we can assume that that patient has tuberculosis. If you remember, Mycobacterium tuberculosis has a very slow generation time of 24 hours or so. So we can bypass the actual need to grow the organism and use the staining technique and the probe technology, and where you see this red amplification, that would indicate that this patient has tuberculosis. Genetically engineering eukaryotes is done through a similar process that we mentioned with E. coli and producing insulin. The process, however, is called transfection. Transfection is the same as transformation, except in eukaryotes. It allows us to genetically engineer plants, such as Bt toxin plants. The toxin is actually from a gene produced by this organism, Bacillus thuringiensis. So we've isolated the gene from the organism and inserted it into plants, and this toxin is toxic to insects. So scientists have incorporated this into corn, cotton, and potatoes, and the actual plant will produce that toxin. So a larva stage organism can try to consume the corn, for example. It will get sick and it will basically stop feeding and die. This is a selectively toxic toxin, so it's not harmful to humans. A lot of people have misconceptions about genetically engineered organisms, and for the most part, you're consuming a piece of DNA that the scientists inserted into a plant, and that plant is producing a protein. So you eat DNA, you eat protein every day. So the effects of genetically engineered organisms um, isn't harmful to humans. Now, in the case of Roundup Ready crops, Roundup is a herbicide that will allow the plant to resist the effect of the actual weed killer. So corn, cotton, soybeans, all types of organisms, sugar beets have been genetically engineered to produce an enzyme that allows it to resist the effects of the actual herbicide. Now, the problem, in my opinion, with genetically engineered organisms would be things that you spray onto the um, actual plant, and that chemical may have adverse effects on humans. Um, the chemical can seep into the environment, potentially into the water source, where again, we as humans may come into contact and it may have an adverse effect on us. The last section of this chapter deals with stem cells. Stem cells are special cells that can differentiate into any other type of cell. So they're called pluripotent cells. And the reason why I talk about this in this chapter is because most of you are going into healthcare, including nursing and dental hygiene, and these stem cells have great potential to be used to replace damaged tissues, um, organs in humans, and potentially cure other diseases. Now, stem cells often go along hand in hand with a controversial subject when we talk about embryonic stem cells. Since embryonic stem cells come from embryos that have not completed the process of determination, embryos must be destroyed to um, get these stem cells. They have great potential for the use in medical applications. So there are ethical issues involved in embryonic stem cells. There are other types of stem cells. There's adult stem cells and iPS cells. Adult stem cells have less potential. They have already went through a process of determination, and they've basically can only become specific types of tissue, so blood, bone, for example. There is no ethical dilemma in obtaining them. However, they just have less overall potential. Still great potential for the applications, but not as great as the embryonic. And then the last type of stem cells are iPS cells, or induced pluripotent stem cells. And it's basically reprogramming mature cells such as an epithelial cell or a fibroblast using a process of transfection. Um, and basically, you make the cell go back in time, I like to say, to become its precursor cells. The problem is the transfected cells contain plasmids with genes that may um, be considered cancer-causing genes. This image is from a paper that was published by a lab that I worked in, expansion and characterization of human embryonic stem cells derived osteoblast-like cells. So what we were trying to do is we were creating these defects in rat skulls here, as shown here in D. We were seeding embryonic stem cells that we had differentiated into osteoblast cells, so bone precursor cells. So we seed these osteoblasts from embryonic stem cells onto these little sponge-like scaffolds, and we insert them into these defects. We sew up the rat, and we harvest them after 8 weeks, and we look for mineralization using an X-ray. So here you can see that there's some mineralization in A, B, and C. D is a control where it's just the defect in the scaffold and there's no cells. So we can use techniques such as PCR and use human-specific primers to show not only that these are from the human source, but we can differentiate them from the rat cells as well. A defect this size in a rat skull will never fully heal. So there might be some healing around the margins here, but it will never fully heal. Now, one of the things when you are differentiating embryonic stem cells. Here's a image showing this is our osteoblast. So this is the cells that we're interested in growing. They're very boring cells. They don't move or anything like that. So when we're differentiating them, if we aren't careful, we can get spontaneous differentiation of these embryonic stem cells. So here we have a mass of cells here. So I'm trying to center onto them, and you can see what's happening is these cells are randomly differentiating into heart muscle cells. And there's some pacemaker cells which is causing these cells to rhythmically contract. So every so often these cells contract. And this is just in a large petri dish. It kind of looks like a smushed 2-liter bottle with a flat bottom, and we find these cells contaminating basically our stem cell flasks. So what we have to do is we have to remove these because these are not the cells that we're interested in actually growing. There is will be a PCR video that was uploaded to a YouTube link, so you can watch the process of PCR as well.